Hi, I'm Akshaya, Iyer, a PhD student in the Department of Chemistry working under Professor Alexander Greer. And today I'm excited to talk about my research on a photochemical principle known as Semi-Curtin Hammett principle, which is all about controlling oxidative reactions using light. So what happens when light meets chemistry? Photochemical reactions are the reactions driven by light. This opens up pathways that normal everyday reactions cannot. Here you can see how light can interact with cells in PDT where reactive oxygen species react with the cancer cells to kill them. Now let's dive into the principle behind my research. Now imagine yourself standing in a valley and choosing which hill to climb based on which one is easier to reach the ground. Did you choose the shorter hill? Well, most of us would, as you will require less energy to climb up and down to reach the ground. Similarly, the Curtin-Hammett principle tells us that when two different molecules are in equilibrium with each other, the way they react depends more on the energy it takes to reach the reaction transition state than on the relative amounts of the molecules. So, A would reach C via shorter transition state. We are specifically studying a molecule called fluoroglucinol, which plants produce as a defense mechanism. It has antioxidant, anti-inflammatory, and antimicrobial properties. Our goal is to understand how this molecule behaves in two forms, phenol and phenolate, how they react with singlet oxygen. This helps us control the reaction pathway in a way that can either protect or promote oxidative reactivity. To understand these reactions, we are using computational methods like DFT calculations, which help us predict how molecules behave during reactions. This allows us to stimulate and study the reactivity in detail without needing to perform every experiment physically. When the phenolate form reacts with singlet oxygen, it forms isohydroperoxide intermediate, which further undergoes protonation. And since there is an abstraction of proton, we refer to it as antioxidant pathway. Whereas phenol molecule first forms a per-epoxide intermediate, which is stabilized by hydrogen bonding. This further undergoes epoxidation, that is oxygen transfer, and thus we refer to it as pro-oxidant pathway. In conclusion, our work outlines two key pathways, one antioxidant and the other pro-oxidant, and I'll show you the corresponding positions in the energy diagram. The antioxidant pathway begins with the phenolate anion, whereas the pro-oxidant pathway with phenol form. The intermediate in antioxidant pathway is isohydroperoxide intermediate, and in pro-oxidant it is peroxyanion intermediate. The isohydroperoxide intermediate undergoes protonation and yields dihydrobenzofuran hydroperoxide, which further converts to dihydrobenzofuran alcohol. In the pro-oxidant pathway, the peroxyanion intermediate proceeds via epoxidation and leads to the formation of stable hydroperoxide product, which amplifies the oxidative reactivity. Finally, I would like to acknowledge and thank NSF Brooklyn College, Graduate Center, my PI, and my labmate Lloyd, and NanoBioNYC for giving me the opportunity to present my work today. To close, I'll leave you with a thought from Alberts and Georgi. Research is to see what everybody else has seen and to think what nobody else has thought. Thank you.